and we're going to look at cellular respiration, which is harvesting chemical energy from organic molecules. It is going to be an example of an exergonic pathway. We'll look at it in quite a bit of detail. And when we look at this pathway, we're really looking at half of the carbon cycle. So if you look at the bottom part of this diagram, we will be starting with organic molecules. In our case, it's going to be glucose, which is an example of a carbohydrate, but you certainly could start with some other organic molecule like a protein or a fat molecule. So if you think of all of those things that we consume to get energy, you could start with those. We're going to utilize oxygen gas from the atmosphere, and these molecules will be broken down with cellular respiration to yield carbon dioxide and water. Now the carbon dioxide that we yield from this is just a waste product that will be exhaled back to the atmosphere. But the importance of this process is that it is going to provide us with energy that we need to produce some ATP. So we will be coupling the exergonic cellular respiration with the endergonic synthesis of ATP. Now the top part of this diagram shows how we get inorganic carbon dioxide, back into an organic form. That process is photosynthesis, and that will be the topic of chapter 10. So in catabolic pathways, molecules are broken down. Specifically, we're going to look at organic molecules that are broken down. And the major pathway that we'll look at is cellular respiration. Cellular respiration is the most prevalent, most efficient catabolic pathway that we know of. And in this process, we will be consuming oxygen gas, which is O2, and organic molecules. The whole point of cellular respiration is to release chemical energy from organic molecules and then pack it away into ATP molecules, which contain an ideal amount of energy for running single reactions. A similar pathway is going to be called fermentation, and fermentation we'll look at the end of chapter 9. Fermentation a lot of times is called anaerobic respiration. And it's called anaerobic respiration because in this case oxygen will not be used, and because it's not used, it will be just a partial breakdown of sugars. Because it's a partial breakdown, the energy will only be partially released, and it will be much less efficient than what we have with cellular respiration. As we begin to talk about cellular respiration, we need to introduce what are called redox reactions. Redox reactions are reactions where electrons are transferred from one reactant to another, and this is going to take place by two different reactions, oxidation and reduction. The oxidation part of it will be that a substance loses electrons. Whenever a substance loses electrons, we will say that it is oxidized. When it loses those electrons, another substance is going to gain the electrons, and we will say that that one is reduced. So oxidation and reduction reactions go hand in hand with each other. One substance will be losing electrons, the other will be gaining electrons, and so when we fuse those two terms together, we just call it a redox reaction. In cellular respiration, we will have many redox reactions taking place. This example that we see right here is the same example we looked at in chapter 2, and it is an example of a redox reaction. We have sodium, which is not very electronegative, and we have chlorine, which is highly electronegative. And when the two are close together, the chlorine takes an electron from the sodium. Now, as we described it before, the sodium turns into a cation or becomes positively charged, and the chlorine becomes negatively charged or turns into an anion. Well, in this redox reaction, we could also use the terms oxidized for the sodium because the sodium is losing an electron in this process, and the chlorine is gaining an electron or becoming reduced. So notice that one thing is becoming oxidized, one thing is becoming reduced. Those two, oxidation and reduction, they will both be taking place in the reaction. Now in this example, we see an electron being completely lost or transferred from one substance to another. Redox reactions may also involve a change in the degree of sharing of the electrons. If we look at this slide here, we see that we've got methane, which is a carbon surrounded by 4 hydrogens, and all 4 of those covalent bonds are going to be nonpolar covalent bonds. In these nonpolar covalent bonds, the electrons will be equally shared by the carbon and the hydrogen atoms. Notice the distance in this diagram that those electrons are from the carbon atom. Keep in mind that the distance from the nucleus that an electron is correlated with the amount of energy that it contains. If you look at the product side of this, if we take another look at the carbon, notice that the carbon now has the electrons held much further away from it. So in a way, the carbon has lost its hold on those electrons, or it is not holding those electrons as tightly. We could use the term oxidized for the carbon now. And the oxygen gas, if we compare the electrons here when they're in O2 with the electrons when around the oxygen and the water molecule, the oxygen is becoming reduced, or it is gaining electrons. So do be familiar with the fact that electrons do not have to be completely lost or completely transferred from one molecule to another. We can use that term redox when we change the degree of sharing of the electrons. Whenever an electron is pulled from an atom, energy is going to be needed to do that. And electrons will lose potential energy when they shift to a more electronegative atom. So if we take electrons from carbon and we move them to something like oxygen, which will be much more electronegative, those electrons will be losing energy. If we draw a picture of this similar to what we looked at in Chapter 8, this would be my free energy on this side. If we take electrons in carbon, these are going to be very high energy electrons. If we take those electrons and we move them to oxygen, those would then be low energy electrons. So these electrons are going to go from having a high amount of energy to having less energy, and that is exactly what an exergonic reaction is. So when this takes place, energy will be released, and then that energy that's released could be utilized to do some work inside the cell. So in cellular respiration, we will be taking high energy electrons and converting them into low energy electrons, collecting the energy that is released and using it to actually make some ATP. So redox reactions are going to take electrons and they will be moving those electrons closer to oxygen. In the process, release some energy that can be used to do work. If we look at the equation of cellular respiration right here, you should, by the time you finish Chapter 9, be able to certainly recognize this equation, and not only recognize it, but be able to identify what's getting oxidized in the process and what's getting reduced. So in cellular respiration, we will be starting with glucose, although you could certainly start with other molecules. Glucose is going to be what we call organic carbon. It is organic because we have carbon and we also have some hydrogen there associated with it. When we look at carbon dioxide is inorganic carbon. Notice that now that carbon does not have any hydrogens associated with it. Whenever you have an organic molecule, that organic molecule is going to have a lot of carbons that have covalent bonds to hydrogens. And those covalent bonds are all going to be nonpolar covalent bonds, which means that these electrons will be equally shared between the carbon and the hydrogen, and those will be high energy electrons. These are high energy electrons, and that is exactly what we're interested in when we go through the process of cellular respiration. So in this process, the glucose, or the organic carbon, is going to lose electrons. And when it loses electrons, it will become oxidized. And in the process of becoming oxidized, it will turn into carbon dioxide. Now, in this equation, you don't see the electrons actually written in there, but if we follow where the hydrogens are at, you can get an idea of where the electrons are at. Keep in mind that a hydrogen ion has a positive charge to it, and those hydrogen ions will follow around the electrons which have a negative charge. So notice that when the organic carbon, when that carbon has its high energy electrons, it also has a lot of hydrogens on it. And when it does not have the high energy electrons, it does not have the carbons in it. So that's when we see it present as carbon dioxide. If we look at the oxygen in this equation, the oxygen gas is going to gain electrons in this process. And when it gains electrons, it's going to then turn into water. Again, if we follow the hydrogens, we can get an idea of where those electrons are at. Notice that once the oxygen gas gains the electrons, it will be in the form of a water molecule. In this process, if we go back to our energy diagram, we will be taking electrons in an organic molecule, so electrons in glucose. We are going to pass those to oxygen to become electrons in water. And when that takes place, it is an exergonic reaction and energy is released. So notice here that we do have energy on the product side of this equation. This is an exergonic process. It does have a negative delta G. And we're going to use that energy to make ATP. So we will use it for an endergonic process. And by endergonic process, we're talking about putting ADP plus phosphate and turning that into ATP. Now, glucose, as we said, is not the only thing that we can use for cellular respiration. We could certainly break down other molecules, but the keys with these other molecules is that they are going to be organic molecules that would be broken down. So if you have other organic molecules, such as those molecules we talked about in Chapter 5, different lipids, proteins, carbohydrates other than glucose, all of those could be broken down by cellular respiration. If we look at the molecules that are being shown on this slide, this first one represents a triglyceride. The middle one is a disaccharide. And the last one would be an unsaturated fatty acid. Notice first off that they are all organic molecules, and in addition to that, there are a number of bonds that are carbons sharing electrons with hydrogens. If you consider that high energy electrons will be held between the carbon and the hydrogens, then the most valuable molecules energy-wise would be the ones that have more hydrogens packed onto them. So if you compare these, the triglyceride would certainly be more valuable than the disaccharide would be. So when we talk about glucose, it is a carbohydrate. It is valuable in that there are high energy electrons held within that molecule, but it is certainly not going to be as valuable as a fat molecule or a fatty acid would be. Now, as we go through cellular respiration, we said we are going to oxidize the glucose, or in other words, we will be collecting energy from the glucose in the form of electrons. We will be collecting them and ultimately passing them to the oxygen gas, but it is not going to be an immediate taking from the glucose and passing to the oxygen. In that equation that we looked at, there's actually about 30 steps buried in there. So there will be a lot of in-between handlers of the electrons. And the electrons that are collected from the organic carbon will first be transferred to a molecule called nicotinamide adenine dinucleotide. We say that this molecule functions as an oxidizing agent because it is going to be causing the oxidation of the glucose. Now here's a look at the structure of this NAD+. And although you do not need to memorize the structure of this molecule, notice that it is a rather large molecule. That will become important later in the chapter when we talk about passing electrons across membranes. NAD+ is able to collect two from the organic carbon, and when it does collect two electrons from the organic carbon, and when it does collect those 2 electrons, it will also pick up 2 hydrogen ions from solution. When it does gather these, that means that it itself is becoming reduced. NADH is what we call the reduced form of NAD+, and in that form, it is holding high energy electrons. Because it is holding high energy electrons, that is a valuable molecule. As we count up energy yields for cellular respiration, we will want to keep track of any NADHs that we happen to have, because they're holding on to the valuable part of the glucose that we will use later to make a bunch of ATPs. Keep in mind that this is a reversible reaction. This NAD plus NADH is a temporary holder of the electrons. It will do the initial collecting of the electrons, but later we will pass them on to some other molecules, and ultimately those electrons will go to oxygen gas. So they're not going to stay here forever. If NADH passes those electrons on to another molecule, it will be oxidized, and in that manner it will turn back into NAD+. NAD+ will do most of the initial collecting of the electrons. However, we will have one other molecule that does collect a few electrons. The oxidized form of this molecule is FAD, and its reduced form is going to be FADH2. When FAD gains high energy electrons, it will gain 2 of them. Then it does turn into FADH2. So FADH2 is the reduced form. It's also what we call the energized or the valuable form of the molecule. Again, it's a temporary holder of the electrons. They will certainly go on eventually to the oxygen gas in this process. Now, in cellular respiration, we said there is going to be a lot of redox reactions in there, so about 30 individual steps, and we will take the electrons from the glucose, which will be very high energy electrons, and we will pass it to a lot of individual molecules before we ultimately give it to the oxygen gas. This picture here illustrates why we're going to use so many handlers of the electrons. Those electrons in the glucose will be very high energy, and the electrons, when they go to oxygen, are then going to be drained of energy. So they're going from a very high amount of potential energy to a low amount, and it's an exergonic reaction, and if all of that energy is released at once, that is basically an explosion. So we would have a lot of that energy being converted into heat energy and light energy all at once. And essentially that wastes the energy because the cell wants to carefully harvest this energy to utilize and pack away in ATP molecules. In this case, that energy would not be used to make ATP molecules, and it would be quite destructive for the cell. So to avoid this, we will have molecules that initially collect the electrons, and then we will pass it to a bunch of other molecules that handle the electrons, and eventually they will go to the oxygen gas. So NADH, the reduced form of NAD+, will pass the electrons to an electron transport chain. Now FADH2, which is the reduced form of FAD, will be doing essentially the exact same thing as well. What the electron transport chain is it is an assembly line of molecules that will pass the electrons one by one to each other, and eventually at the very end of the electron transport chain we will have the oxygen gas that gathers those electrons. The whole purpose of the electron transport chain is to ease the transition of those electrons from high on the energy diagram to low on the energy diagram, and basically get them there in many steps, which will then avoid that one explosive reaction. This will enable the cell to capture that energy and use it to actually make the ATP, which is the whole purpose of cellular respiration. We want to carefully collect the energy and use it to make ATP. So if we look at this diagram again, this more closely reveals what we will be doing in cellular respiration. We will be taking electrons from our food, from glucose in the example that we're going to look at, and we're going to have those electrons collected by NAD+, which then turns into NADH. NADH will carry those electrons to the electron transport chain, which will consist of many different molecules which are at varying levels on this energy diagram. NADH will pass those electrons to the first member of the electron transport chain, or the first member of the assembly line. And when those electrons are passed to that first one, that is an exergonic reaction, and we will have an amount of energy released. The cell will then attempt to capture as much of that energy as it can. Then those electrons will be passed to yet another member of the electron transport chain, and energy will be released, and the cell will attempt to capture it, and so on, until eventually those electrons are passed to oxygen gas, and oxygen gas will pick up some hydrogen ions at the same time, and it will become water. Notice that with each step in this electron transport chain, you have basically a small amount of energy that is released. That would be the delta G for that reaction, and it is going to be a negative delta G at each step. Okay, so we have lots of little bits of energy that are being released along the way. Because it's little bits of energy that is released at a time, the cell is going to be much more efficient in the collecting of that energy and then ultimately packing it away in ATP molecules. Notice that overall this is most definitely an exergonic reaction. Notice that we do have a negative delta G. So we will be releasing energy, and then we will be using that energy for the endergonic synthesis of the ATP molecules. If we look at where the electrons are going in this whole process, we will be taking them ultimately from some kind of food. That food is going to be an organic molecule. So we will take them from the organic molecule. They will be collected by NADH and FADH2. Those molecules are doing the initial collecting of the electrons, but they are temporarily hanging on to them because ultimately they will pass the electrons to the electron transport chain, and at the very end of that electron transport chain will be our oxygen gas, which is O2. Lots of redox reactions involved here because every time we transfer or pass the electrons from one molecule to the next, that's reduction and it's oxidation taking place as well. And inside this, there are lots of exergonic steps because every time we pass them, we're going to be releasing little bits of energy. Now, if we talk just an overview of respiration, the process of cellular respiration is going to be a cumulative function of 3 individual metabolic stages. We'll talk about the overview here, and then later we'll go into the individual details and enzymes that are involved in these steps. The first stage is going to be glycolysis. The word glyco means sugar and lysis means splitting. So just the term there means splitting of sugar or cutting a sugar. And that is pretty much exactly what we're going to do. We will have 10 steps involved in that splitting process, but we will be taking glucose, an organic molecule, and essentially splitting it in half. So we will end up with 2 molecules of pyruvate, which will also be organic when we finish glycolysis. Glycolysis is going to take place in the cytosol, and the purpose of glycolysis is really just to start the breakdown. So we will be starting the breakdown, and by starting the breakdown, we are beginning to remove the electrons from the carbon. If we are beginning to remove the electrons from the carbon, keep in mind that we are then making NADH, because NAD plus is going to go and gather the electrons and it will turn into NADH. At the same time, we will make a little bit of ATP. We'll actually get 2 ATPs profit from glycolysis. It's not a whole lot at that point. So glycolysis is really just getting the process started. The second stage of respiration will be the citric acid cycle, which is also known as the tricarboxylic acid cycle or the TCA cycle. We can also call it the Krebs cycle. So depending on which textbook you look at or which website, you may see it referred to in a different manner. Your textbook tends to call it the citric acid cycle, so we'll try to use that terminology. And what the citric acid cycle is going to do is it is going to complete the breakdown of the glucose. By complete the breakdown of the glucose, this means that the remaining high energy electrons will be removed from the carbon. That means that we will be making even more NADH, and we're also going to see some FADH2 made here as well. So we collect the high energy electrons. We will also make a little bit of ATP. It's going to be another 2 ATPs that gets made in that stage. And that second stage is going to take place in the mitochondrial matrix. That's the very inside compartment of the mitochondria. So with the first two stages, the job of those is really to collect the high energy electrons. We will make a little bit of ATP along the way, but really it's pretty small in the grand scheme of things. So each of those stages makes about 5% of the total ATP that we're going to make. The major goal, though, is to collect the high energy electrons. By the time we finish the first two stages, the organic carbon will be gone, so we will have completely oxidized that and turned it into carbon dioxide, which we then exhale to the atmosphere as a waste product. The third stage is called oxidative phosphorylation. In the third stage, we will make 90% of the ATP that we're going to get from this process. So the third stage is by far the most valuable of all of the phases. This is the stage where we will actually collect or release the energy from those electrons that we collected previously. This means that this is the point where we're going to actually move them down that energy diagram. So take them from the high energy state down to the low energy state where they will be when they are handed to the oxygen gas. This last stage is going to occur in the mitochondrial inner membrane, and that mitochondrial inner membrane can also be called the cristae. When we discussed mitochondrial structure in chapter 6, we said that the inner membrane was folded because that would then increase the surface area of that membrane. Notice that if we increase the surface area of that membrane, we will be able to run more of this very productive third stage of respiration. If we look at this process in regards to the cellular structure, we will be starting with glycolysis in the cytoplasm. Glycolysis will take a glucose, and through many steps, we will split that glucose into two pyruvate molecules. When we do that, we will be collecting some of those high energy electrons from the carbon, and those will be passed to NAD+ to make NADH. Notice that because glycolysis takes place in the cytoplasm, the NADH will be produced in the cytoplasm. Then we are going to take the pyruvate. It will be shipped into the mitochondrial matrix because that is where the citric acid cycle will be taking place. And pyruvate is an organic molecule. And since it's an organic molecule, there's still a lot of high energy electrons inside that molecule. The citric acid cycle will take place in the matrix. In that cycle, we will remove the remaining high energy electrons from our organic carbon. And that means that when we finish the citric acid cycle, all of our organic carbon will have been converted into inorganic carbon, which will then be exhaled or released to the atmosphere. As we remove the high energy electrons, we pass them to NAD+ to make NADH. We also pass some to FAD to make FADH2, and those are going to be inside the matrix of the mitochondria. The third stage of respiration is going to go to the electrons that were previously collected. Notice there are some in the matrix of the mitochondria and there are some in the cytoplasm of the cell. The vast majority of them will be in the matrix of the mitochondria. The third stage of respiration, the oxidative phosphorylation, will take place on that inner membrane of the mitochondria, and that one is going to utilize those high energy electrons, extract the energy from them, and ultimately pass them to oxygen gas, which then turns into water. Oxidative phosphorylation really has two halves to it because we will be running the electron transport chain where the energy will be released from the electrons, and then we'll have the second half, which is going to be called chemiosmosis, and in that process we will be taking the energy that is released and we will be using it to actually make the ATP. We will in cellular respiration have two different methods of making the ATP. The first two stages, glycolysis and the citric acid cycle, together those are going to make about 10% of our ATP. That 10% of ATP is going to be made by the process of substrate level phosphorylation. In substrate level phosphorylation, we will have an enzyme that is called a kinase enzyme. So Kinase. This kinase enzyme is going to transfer a phosphate molecule or a phosphate group from a substrate onto the ADP to yield ATP, which is exactly what we want. That is an end product of respiration, and that can then be used inside the cell wherever we happen to need some ATP. So in substrate level phosphorylation, we are simply transferring a phosphate from a substrate molecule onto ADP to yield ATP. We do want to keep in mind that it is a kinase enzyme that is going to catalyze this reaction, and that this is taking place in both glycolysis and the citric acid cycle. For every molecule of glucose that we start with, and we completely break down through this process to carbon dioxide and water, the cell will make up to about 38 molecules of ATP. Now we say up to about 38 because there is a little bit of variation in there, and we'll talk about where that variation comes from. But if we look at the efficiency of this process, in one glucose molecule, or in 1 mole of glucose, there are 686 kilocalories of energy. We're going to take that 1 glucose and we will make 38 ATPs. There are 7.3 kilocalories of ATP and of kilocalories of energy in each mole of ATP. So if we talk about the efficiency of respiration, we're going to take those 1 glucose and we're going to make 38 ATPs, which we said are 7.3 each. What that means is that if we take this 7.3 and we multiply our 7.3 times 38, we're going to get 277.4 kilocalories packed away in ATPs. We'll notice that 277.4 obviously is not 686. That means that we are not getting all of our energy packed away in ATPs. So if we look at the actual efficiency of it, if we take the 277.4 and you divide that by your 686 that we have, you get an overall efficiency of 40.4%. So cellular respiration is 40% efficient. What that means is that when we break open that glucose molecule, we will get about 40% of the energy packed away. Now keep in mind we said that the first law of thermodynamics says that energy cannot be created or destroyed. Okay, so the other 60% is not just disappearing here, it is being converted into a different type of energy, such that it is not found in our ATP molecules. The vast majority of that other energy will be released as heat. These will be small amounts of heat that are released along the way. So cellular respiration can certainly be used as a way to generate heat inside a cell or inside an organism for that matter. Cellular respiration, while 40% may not seem that great for efficiency purposes, 40% is the most efficient process that we know of. So we do consider cellular respiration to be highly efficient and the most efficient catabolic process that we are aware of.